Good afternoon, everybody. Thank you all for joining. It is a little after noon. We still have a few participants that are joining. Just as a reminder, this town hall is being recorded, and we will have a video link that will be posted on the COVID-19 BioBank website, which you then can share with colleagues who are unable to attend, or if you missed a portion of it, it will be housed on the website and it is being recorded. Please note, we will go through the slides. There are several presenters today, and we are holding Q&As at the end. But in the interim, if you have any questions, please feel free to type them in the Q&A box that is at the end. And we will have a moderator who will be working with the audience, the panelists, to address those questions as they come in. And with that, we will begin. I'd like to introduce Dr. Thomas Flaig, our Vice Chancellor for Research at the university.
Well, thank you very much, Natasha, and it's great to have all of you joining us. Just looking down, it's about 185 people on the call. So let's just look at the agenda, but I'm glad you're here, and I hope we can provide you with some useful information and also just acknowledge all the many people that have contributed to this project in short order on kind of an emergency basis. I think it's very pleasing to see how this has come together, and I hope it's a great resource for you. So I'll make a few high-level comments. Adrie van Bokhoven from the Biorepository Cohort will talk about their efforts, really in a central role in assembling these samples. Bryan Haugen's going to talk about the Steering and Allocation Committee. Bryan chairs that committee and has done a fantastic job of providing a nice structure for this process. Matt Steinbeiss will talk a bit about the project management and some of the operational aspects of the biobank. Tel Bennett from the CCTSA is going to talk about the clinical correlation. Been doing some wonderful work with that, and it's going to come through. And Joaquin Espinoza is going to speak a bit about the approach they used in the Linda Sternick Center for the Trisom Explorer, some of the learning he did to really share data very effectively. How we're going to try to bring over some of that to the process here as well. That'll be followed by some questions and answers. And again, as Natasha's indicated, we've got about 200 people out, so just use that Q&A box at the bottom. Next slide, please. So again, I want to welcome you. Thank you for joining us. Our goal today is to give you some concrete information about the biobank, how it works, what's in the biobank, what our future plans are, and how to access it now. You know, I think all of us were struck a few months ago by the extraordinary times which we encountered. If you'd asked any of us 6 months ago, what are your plans? It would have been to shut down a large portion of our research operation and adapt to work from home on a large scale, nationally, internationally. So it's been an intriguing, challenging, difficult time. What I think a lot of us realized, obviously we made changes in the lab operation. We've then been able to ramp up over time. We've done the same thing with both clinical and community research. But it became clear that for many people who want to research COVID-19, they would need access to samples. It also became very clear that our access to the clinical care areas would be extremely limited. I think one of the initial observations we made is there were maybe a half dozen groups that were contemplating or actually were starting to put in requests for different biobank proposals. And, you know, I think we've traditionally just sort of had a decentralized process where everybody can, you know, obviously would do their own thing. In a special circumstance, we said, let's try to do a single institutional effort. Let's put together our best resources, have one consent, one process rather than a myriad of these. And especially considering the difficulty in patient access areas. I've had a chance to do and lead a lot of different clinical trials over time. I've never had to think through how to do this without being able to access patients, how to do it remotely, how to identify patients remotely, consent them remotely, all sorts of different confounding factors that came into this. And so we got together a wonderful group of people that helped to think through each of these different problems to get us to the current state. So we essentially launched this institutional effort as a central resource for the university. Next slide, please. And again, we're going to talk a bit about the thank yous. To me, it was really heartening to see many different entities and individuals across campus. We mentioned the Biorepository Cohort, the HIMSR shared resource, the clinical labs at both hospitals, the committee that came together, regulatory affairs engagement, CCTSI, in many ways, the School of Medicine Information Technology Group, providers treating COVID patients. So we called providers and said, how can we do this? A variety of people in the emergency department, on the hospital floors, at Children's Hospital, all stepped forward and said, well, this will work, but that won't, and that typical thing won't work, and so forth. Investigators who have been patient as this process developed, I think it's moved about as quickly as we could in these circumstances, but there's been a lot of patience. And of course, to the subject participants and the patients who have received a call in their hospital rooms frequently saying, I'm a coordinator for a biobank, would you like to learn more about it? And that's an unusual way of doing clinical research. Next slide, please. So again, the last slide for me will transition to really the substance of our talk today. The goal of this biobank was to collect these samples from patients who are suspected of the virus. We have patients that had this in their differential and were negative, and patients that had the differential were positive. So we've got some controls in there as well from campus. So it's a collaboration between the university, Children's Hospital, and the adult university hospital. The committee, which is chaired by Bryan Haugen, as I've mentioned, both serves a steering function and allocation function, has been very thoughtful in looking at the requests that have come in to date. I will note, and Adrie and other people will comment on this, we have a prospective sample. We'll see the numbers today. We've got a decent number of patients who are prospectively consented, and then have very carefully processed samples. In some cases, we're able to get longitudinal samples. And then the biorepository has done a great job of collecting from the clinical labs discarded samples. So again, a large number of samples in that regard. We are asking investigators to use this resource to share those samples. Share those results. So if you run a certain assay on these samples, we're trying to make a biorepository in data such that we can share those with a variety of different people. The clinical correlation then comes in from Health Data Compass, so using a very robust clinical resource. And we're now in the process of starting to fulfill some of these sample requests. And so this seemed like a good time to have a town hall to address the questions and so forth that you may have. Next slide, please. So with that, I'll hand things over to Adrie van Bokhoven. We've talked a bit about the agenda. We as speakers are going to try to keep our comments to about 35 or 40 minutes to allow plenty of time for questions. And as we've noted, please use the question and answer at the bottom. So again, I have sincere appreciation for Adrie and what he's done in the biorepository. Adrie, please take it away.
Hey, thank you, Dr. Flaig. Next slide. So, like Dr. Flaig said, so we're doing this at UC Health and Children's Hospital. And we have two routes of getting specimens. One is through patient consent, where the coordinator teams, both at Children's and UC Health, approach patients by phone, consentees, for doing a research collection after consent. And then we have the waiver of consent, which pretty much means anybody that has a COVID test, positive and negative, we can try to pull in clinical specimens. And most of these specimens come from the clinical lab at both institutions, and these are really random specimens that are no longer needed for clinical care. All these specimens, both of these are flowing to the biorepository facility for management and coordination, and specimens processing needed, and that's the case for the research collections. They move real time to the Hansel lab, which set up a BSL-2 plus facility actually handle these specimens early on in this process. So, and after processing, these all flow back into the biobank at the biorepository facility. So really inventory and move all the data on board, where also patients are in. It's linked up with the patient information, and the data can be moved to Health Data Compass. And looking on the right section of the slide, which explains a little bit what the other speakers are going to talk about, how research requests are done, how they move through the steering committee, and how with Health Data Compass, based on the clinical data, we can do a cohort selection, making sure specimens are available, and then serve research projects. Of course, I have the honor to present these, but there are a lot of different individuals and entities involved in this. For most, some cancer center shared resources like HIMSR, the pathology shared resource, which actually formerly was the tissue biobank histology shared resource. We actually just changed our name because we merged together with molecular path. And then also coordinator teams, and especially the clinical labs at both UC Health and CCO. We couldn't have done any of these arms without these two labs. Next slide.
And Adrie had just a little bit of audio issue there. It sounds like it's back. So I may interrupt you if we lose your audio again, but we can hear you fine right now.
Okay, is this better?
This is better right now. Yeah.
Okay, so let's talk a little bit more about the prospective biospecimens. So we have collection kits in the clinic at a few of the floors, and after the coordinator approached the patient and the patient agrees to consent, a draw is ordered into EPIC, a research draw, and these samples are collected throughout the day or early on in the morning draws, or also sometime in the weekend. We're actually collecting 7 days a week for this protocol. These samples are moved to HIMSA for processing, and in these 3 different tube types actually lead to 6 different isolates. 2 different plasmas, also PBMCs, viable cells. We isolate between 20 and 30 million cells of these. We can do Bokhoven, which we fix, and also red blood cells. So there's a number of analyses possible, and Dr. Espinoza later on will go into detail a little bit why we kind of tailor this collection kit to these tubes and these isolates. As said, these are coming from UC Health and Children's. And early on, we only did a baseline collection just because things were challenging, even to get specimens out of the clinic. We couldn't really do that without moving to the clinical route to the clinical lab. And baseline was already challenging, but later on, we actually brought in another collection opportunity for longitudinal specimens for when the patient is longer in the hospital, so we can collect between 3 and 7 days, or even longer, 2 weeks to 3 months. This doesn't capture the full scale of specimens that might is needed for research, so we can also supplement these with remnant clinical samples, which we capture through the clinical route. Next slide.
Adrie, it looks like you're muted now. Adrie may be having some audio problems. I'm just going to talk, and Adrie can jump back in as he can. So again, there's 2 kinds of participants. Adrie, feel free to jump in. Both those that are prospective and consented, again, has that rich processing data that Adrie just showed, and those with a waiver of consent. There's a large number of remnant samples that Adrie and the team have been able to collect. And again, these are any samples that are basically discarded from the clinical side of things between 1 day, in some cases, even months afterwards. And we've been accepting about anything that the clinical labs are willing to release. We might have lost Adrie there in that time span. So I'd say nasal swabs over 10,000. And those have come over unprocessed essentially. So there's a large number of nasal swabs. That would include both positive and negative patients. Whole blood, a decent number of those were 1,500. Plasma and serum in the 1,500 to 1,000. About 400 urine samples and FFPE. So again, just to understand, there's this prospective, very well processed samples, a limited number, and then these large numbers of discarded clinical remnants. So Audrey's done great work. Sorry we had some audio problems there, but hopefully we got most of that through. We can go to the next slide. And then we'll just transition now to Bryan Haugen talking about the process, the steering committee, and allocation.
Thank you, Tom. I just wanted to give a few slides about the requests in the allocation committee. The committee consists of 9 people you see here. There are 5 voting members, myself, Dr. Peter Gottlieb, Dr. Eva Grech, Dr. Adrie von Bokhoven, and Dr. Marty Voskuil. And the idea with this group is to make sure we have a broad enough representation with adult medicine, pediatric medicine, basic science, pathology, and clinical research with some specific emphasis in pulmonary medicine and immunology. So whenever we replace members, we try to keep that in mind so we have a broad representation of the institution and expertise. We also have the 4 non-voting members, Dr. Flaig himself, Dr. Allison Lakin, who is our Associate Vice Chancellor for Regulatory Compliance and our Research Integrity Officer, Marcia Finucane, who is our biological safety officer, and Matthew Steinbeiss, who you'll hear from in just a little bit, who's our special projects manager and really a key point person for applications to this committee. Next slide, please. And so the committee charter that was developed and some of the logistics, the biobank was established, as is said, to collect and bank these biological biospecimens, and also to link pertinent clinical data, which you'll hear from Tel Bennett in a little bit, from patients related to COVID-19. Next. And as you know, Tom Flaig is the PI. Next. And one of the purposes of this committee is to advise on policy and processes to facilitate broad access to this biobank resource and database. Next. And another big component we have is to review individual applications for scientific merit and use of the biobank materials. And those are our two key components that we look at on what is the scientific merit and how is the biobank materials being used to their fullest. Next, please. And one thing we did more recently was to assign a liaison or champion for each proposal from that voting group to help facilitate approval. So not only do we try to rapidly send letters back to investigators, but then we have a point of contact person who usually will talk to them on the phone to work through any concerns the committee has to try and get this approved quickly so people can have approval and access to materials. Next. We meet every Wednesday at 7 a.m. To review these proposals or review revisions to the proposals. We used to meet twice weekly, but we meet now once weekly at 7 a.m. And next. This you'll see again at the end. This is the link to where people can go to set up and get their application process moving. And again, they can contact me or any of the members or Matt if they have questions. Next slide. So I would next like to introduce Matthew Steinbeiss, who is the biobank project manager. Matt.
Hi, everyone. I just wanted to take a short amount of time to talk about what will help the biobank application process go smoothly. We would like to see a fully fleshed out project idea, which includes 1 to 2 pages of scientific rationale. This can be from a specific aims page for your protocol, but the main idea is to have the committee fully understand the science behind your proposal. It is important that you describe fully the types of samples that you're requesting, including the number of samples, volume, etc., as well as what type of data you're looking for to be associated with these samples. For example, length of ICU stay, complication, comorbidities, etc. We also request a plan for data sharing. As this is a community effort, the data generated for your research is expected to be shared back to the research community via the steering committee. And we can work with you on the data sharing aspects of your protocol during the application process. We want to make sure that lab safety has been well thought out, as well as your timeline for starting, performing, and completing your research. The REDCap link is below, but can also be found on the BioBank webpage. Next slide. The webpage was mentioned earlier, but I wanted to draw special attention again as it's being updated regularly with new information. For example, this town hall, the FAQs are regularly updated as well as other information. So it would be a good idea to have it bookmarked. My contact information is in the FAQ section, so I am available to answer any questions you may have about the biobank, the application process, or I can connect you with the right person if I'm unable to answer your questions. Next slide. Next, we're going to hear from Dr. Tel Bennett, the director of the CCTSI Informatics Core, who is going to speak on the clinical data coordination of the COVID biobank.
Thanks very much, Matt. So this slide shows the 5 main data sources that are integrated in the core data mart. So the core data mart sits on the Compass Data Cloud, so in the Google Cloud environment that they have established. And the Compass team does the functions that you see in the middle of the slide, integration and linkage. They serve as the honest broker for the process, and some of the analytic variable creation. I'll talk more about that in a second. Then the 5 main data feeds, starting at the top, you have the links to the biospecimens that Bryan and Adrie and Matt talked about. These are accessed through the link to the Encore system. And then on the two sides, you have both direct links to EHR data that will be updated regularly from both UC Health and Children's. And this is a very broad EHR data pool, as well as instruments based in the CCTSI REDCap instance, where information that's not usually, and concepts that are not usually collected well in structured EHR data, and these would be things like pre-hospitalization symptoms or exposures. On the UC Health side, this is a large chart abstraction project about inpatients led by Christine Erlinson and Lisa Schilling and Shanta Zimmer, and conducted by a large cadre of medical students. On the children's side, this is largely collected by a parent survey, but has similar data elements. And then the analytic variable creation process has just started. We just got final regulatory approvals, and will be a joint effort between the COMPASS team, School of Medicine data scientist who works for me, as well as the CCTSI Biostatistics and Epidemiology and Research Design course. Next slide, please. So as you know, there are a number of cohorts in the main BioBank protocol. This is the largest one, the waiver of consent cohort, just to give you a sense of scope. It's quite broad. The inclusion criteria are they received any care at Children's or UC Health, and they were tested for SARS-CoV-2. The only exclusion is that they were approached for participation in one of the other BioBank cohorts and declined. And so as of last week, there were 87,861 unique patients in this cohort. Next slide, please. So the process for requesting data, there is an overarching BioBank protocol, but if you want to use the data or the samples, you need an IRB for your study. 2 and 3 should happen in parallel. So the specimen request process that Matt just described, and then we are leveraging the existing robust COMPASS data request process, and the link is right here. And then I put in number 4 here to get across the idea that very frequently engagement with the COMPASS team, and perhaps with the BioBank and the CCTSI team, is going to be needed to really define exactly how, what variables specifically you need, and how to define them in both the clinical data and the links to the biospecimens. One of the big advantages of building it in the way that we have is that data delivery should be much faster than a COMPASS data pull in the regular queue. COMPASS is currently estimating they'll be able to deliver data from this data mart in 2 to 4 weeks, depending on the complexity of the data requested. Next slide, please. So in the future, I'd like to point out just a couple of additional features that we have planned. On the left side, from both Children's and UC Health, you see a box where we see a number of data types. We plan from UC Health and hope to from Children's in the future bring in raw chest CT and chest radiograph images into the Google Cloud environment for COMPASS. These will be the first images that COMPASS will hold, and we're excited about that. Joaquin is going to talk about a large collection of omics data that will ultimately be linked to the clinical data. And then on the right side of the slide, you see a number of visualization and sharing activities. And one I want to point out is this N3C link. This is a national effort. Next slide, please. So the National COVID Cohort Collaborative is supported by a number of federal entities, and both Children's and UC Health are participating. Contributed data will be harmonized to the OMOP Common Data Model on a secure federal cloud environment, and then made available to investigators for analytics. You can't download the data, but you can analyze it on the platform. And so this should be up in the next couple of weeks, and I'll be sharing information with the campus community when it's available. Next slide, please. Now I want to introduce Dr. Joaquin Espinoza. He's the Executive Director of the Linda Cernick Institute for Down Syndrome.
Hey, thank you, Tel. It is great to have this chance to share a little bit with everyone our ongoing activities here to create the COVIDome, and I should credit Ron Sokol with coming up with this fancy name. What is it? A multidimensional data set to accelerate collaborative research on SARS-CoV-2 and COVID-19. Next slide, please. So the goal here is to generate a core data set with key omics data types and clinical data that we can build upon as users of the BioBank generate other data types. So the core data set will be clinical data, and Tel has explained that. plasma proteomics, thousands of proteins on a couple of different platforms. And you can imagine that we'll have a lot of proteins important for the understanding of SARS-CoV-2 infection and the COVID-19 pathology. Plasma and red blood cell metabolomics and lipidomics, deep cytokine profiling by mesoscale discovery assay, whole blood transcriptome from PAX RNA tubes, so that is all RNAs in the blood, and then immune cell maps via mass cytometry. Now, importantly, all data types will be generated from the same blood draws, which means that then you can integrate across these data types for further discoveries. Next slide, please. So the key here is to biobank properly, and Adrie mentioned this on how the samples are being processed. We're going to be focusing for the COVIDome on the so-called prospective samples, not the remnant samples. And these prospective samples are processed by Adrie's biorepository, but also by the team at the HIMSR in the fractions that you see here. So after blood extraction, we can get EDTA and sodium heparin tubes, and then the EDTA plasma is the preferred source for proteomics, lipidomics, metabolomics, and cytokine profiling. Then from the Buffy-coated white blood cells from either tube, we can go into a FICO gradient and get the PBMCs as the preferred source for immune mapping by mass cytometry. Red blood cells can provide a different source for proteomics, lipidomics, metabolomics. And then as you heard from Adrie, the biobank is also collecting PAX RNA tubes that will immediately freeze, if you will, the blood into that state at the moment of the blood draw, and you can later on do a transcriptome analysis. So we have about 100 samples, maybe a little more today, that have undergone this full process. Next slide, please. So the goal here is to eventually integrate all these data sets and other data sets that may be generated by other users of the biobank on the same samples into a central data repository, a relational database, if you will, that will enable different modes of collaborative analysis. One of them would be a research gateway, an online portal baptized as the COVIDome Explorer. But of course, there'll be a devoted team of biostatisticians and bioinformaticians, we call the Deep Blue team, that will do more sophisticated queries. And then ideally in the future, we'll be able to employ crowdsourcing tools outside of the institutions, such as Dream Challenges, powered by Sage Bionetworks, again, to enable collaborative discoveries. So the goal here is that by looking at multiple integrated data types, we can have more discoveries about the architecture of COVID-19 pathology. Next slide, please. So for those in the audience today who are not familiar with omics approaches or with the work at the Cynic Institute, I would like to give you some peace of mind that this is feasible. And what I'm showing here in the left, it shows multi-omics analysis of more than 400 participants with or without Down syndrome. D21 are control participants, D21 is individuals with Down syndrome. And I'm showing you just 4 data features from different omics: elevated interferon receptor from the transcriptome, elevated CRP from cytokine profiling, elevated MMP1 from proteomics, elevated kynurenine from metabolomics. And I picked these because these are all relevant, of course, for an inflammatory condition such as COVID-19. And here in the right, I'm showing you the proof of principle of what a collaborative data effort could look like. If you're not familiar with this portal, I invite you to visit it, the Trisome.org Explorer, where we upload routinely all the data set being generated in our study of the cohort people with Down syndrome, the Human Trisome Project. So we are trying, as Tom Flaig said earlier today, to apply some of the lessons and the expertise developed in the study of people with Down syndrome for the study of COVID-19. Next slide. This is a large effort, and I always, dread these credit slides because I'm always afraid I'm going to forget somebody important. But there's a lot of people that has come together to enable this. The biobank, the biorepository, the HIMSR, Jill and King have been outstanding. TEL in capturing the clinical data, and then the Panomics team, people from the Cynic Institute, Elena's team for CyTOF, Angelo and Kirk for everything that is mass spec based, and then an amazing team of analysts, the so-called Deep Blue team listed there. And that's all I had, Tom. It's back to you.
Thanks so much, Joaquin. And I think it's a great example with the Trisome Explorer of what can be done and is feasible, and to take both molecular scientific expertise and that functional expertise together to help the COVIDome has been just great. And I love the COVIDome, Ron, if he's listening. So, okay, and I might just flip one slide forward. We're going to jump into some Q&A, and Matt's been, I think, trying to correlate through there. Just remind folks at the bottom of your screen, there's a Q&A button. there's about 230 people on, so may just have you type your questions in that way. Just as Joaquin said, I always worry about putting any, there are so many people that have helped this. You just saw a wonderful team of people that came together quickly, all said yes to me, I'd love to help in this emergency situation. And I think we put together a very cohesive core that can be a great resource for our research community. Again, all the different groups listed here have been incredible. There are other contributors as well, again, across both hospitals, across the university. We'd just like to also mention the study participants. So again, imagine if you're sick, you're acutely in the hospital, your phone rings, and that's one of our coordinators through CREST, ask if you'd like to participate at BioBank and receive more information about that. So the 100 or so patients that have said yes to this, we're very much appreciative to that. On the bottom right, you also see the BioBank website. We put a good amount of effort there, the we being Matt, primarily to set that up. And so we hope that's a good resource for you. We would love for this to be used extensively by the research community. So with that, Matt, I don't know if you've had a chance to look at the questions and maybe there's a few thematic things you'd like to bring forward.
Yeah, a couple great questions has come through. I've been trying to answer a few while people were speaking, but I think there's some great ones for the group. One of the first one is, what is the priority given to different projects? For instance, a multi-center funded study in which we are participating versus a single center study that is locally initiated.
So Bryan, I don't know if you'd be comfortable kind of taking that when I maybe just generally talking about the allocation committee. I don't know if we mentioned how many you've actually adjudicated or looked through already and so forth.
Yeah, thanks, Tom. We've probably reviewed and been working on and approved most of them, 20 to 25 different protocols. What we do is we do give it a priority score. Now that said, there's nothing so far that we've said we can't give any material to. The goal is to try to to give appropriate material to everybody. And so we do have a score in case there is some priority as far as especially limited materials. But again, we look both at the science of it. We do look at studies, obviously, that either are linked with industry or maybe already have NIH funding. We look at very favorably on the ones that already have NIH funding. We, again, try to look, though, at what type of material and how much material do they need to accomplish it, to try to get the biggest impact for each protocol. But so far, again, there's been nothing that has been outright rejected. We try to get these all to an approval state.
And again, I'm an observer in those meetings, very formally and thoughtfully done. 1 or 2 protocols typically reviewed in an hour session. And frequently what you'd see is that someone might have requested a mil and maybe 100 microliters or 50 microliters be sufficient. So I think there's a lot of judicious thought about the samples needed and so forth. A good question.
For the next one, are there plans to include other unique sample types, for example, placenta, or should researchers move forward with normal recruitment avenues for such samples? Along with that, I think we should talk about kind of the non-biobank approval process for COVID studies as well.
Well, how's this? Adrie, would you be comfortable? Audrey's done such great work. I know you had a little audio trouble at the end, hopefully at the beginning there. Hopefully it works. But do you want to just talk about maybe, I think someone mentioned a placental sample here. We've had a difficult task of saying we want to serve everybody, but we've got to have enough focus so we can be successful at the core mission. And Adrie, maybe just leave your video off and if you'd like to respond.
Yeah, I can respond to that. I'm sorry, my internet dropped out earlier. Placenta actually is the sample type we will be doing because we just opened up another COVID biobank for pregnant women. So we're retrieving it there. We did retrieve a number of placentas already through the clinical stream. These are all FFP materials. For other sample types, we've gotten requests for certain sample types that are just very hard to cover under this or require special collections at the ICUs and so on. And for those, we usually tell investigators to go their own route, but I would always suggest first come to the biobank with your proposal, see how it fits in. And although we might not be able to help you right away with what we have, we might have suggestions of how you can approach that.
Adrie, I think that's very helpful. And Matt, do you want to add anything else about other approvals around that?
Yeah, I think for the time since the COVID pandemic started, the research community on campus has tried to streamline the process of getting through COMERB and doing COVID-related studies, but there also has to be an approval process to make sure researchers aren't approaching subjects for the same exact study, and that COMERB requests approval from the committee, despite the fact that if you're not going to be using samples from the biobank, that if it is COVID related, that a governing body needs to be aware of the research and to make sure that we're just working together on campus for this type of research.
Thanks, Matt.
Yeah. The next one is, how much serum plasma per patient would researchers be allowed to be obtained?
Yeah, and again, I might just, I'll make a couple comments and Bryan can give the right answer, but I would say that, in general, the prospective samples are quite limited. We're very intentionally going to use some of that for the COVIDome sort of central analysis that'll have that stored data then centrally so that everybody can use it. Those are, I think, precious samples with limited quantities. The remnant samples we've got measured in the 1,000 range. There's more flexibility there, but that's one thing the allocation committee would look at pretty specific. And Bryan, I don't know if you want to comment further.
Yeah, just for the remnant sample, I think what we're trying to do, as you said earlier, Tom, is to just have people request the minimum amount they need to get their work done, and not to try to request excess sample for something else, but for what you're trying to get your work done. So we don't have an upper limit of what somebody can request. We just usually then would go back to the investigator and ask, is this the sort of least amount of material you need to get your work done?
Right.
The next one, I believe this is for Joaquin. Is there any genomic analysis being done on these samples?
Great question. So we have the intention, it's not part of the core COVIDome at this point, but there is a possibility of collecting DNA and partnering with larger enterprises that are looking at genome variants and their impact on COVID-19 pathology. Our study here will not be power enough probably for identify genetic variants that may have an impact, but we could contribute to other multi-site efforts that will be more power in that regard. So it's definitely within the realm of possibilities, not listed right now as a core COVIDome data type.
And I just added that I think there's about 1,500 samples with whole blood, so there's a fair amount of DNA there, but I think Joaquin's framed that very well.
Alrighty, and then, so what, if any, community engagement process is in place overall and in special projects? So a question about community engagement. I might ask to get a little bit more clarification.
Yeah, so happy to think through that. Maybe if they just want to give just a little bit more information there. I mean, one of the challenges of the situation, again, I've done a decent amount of clinical research, is trying to do this in the pandemic setting, not be able to, interact with the patients directly. Even, the great help we've had from the emergency department, from the hospital floors of both hospitals. Without their help, we wouldn't even be able to operationalize it with the, the local nursing staff and so forth. So I think engagement in general has been a real challenge. I'd be happy to think through more aspects of that if you want to add some more.
So there was also a question, I don't know, Adrie, if your internet connection is better, if we could review kind of the slides that were cracking up maybe towards the end. But one of the specific questions was, what are the numbers for unique pediatric samples versus unique adult samples? I believe those might have been on Adrie's slides, but I am not positive.
That's a good suggestion.
These numbers were actually, Matt, these numbers were not on the slide, but certainly for the adults, that goes into the thousands. For the pediatrics, we're really talking about numbers under 100 because the patient population is with COVID is much less at CCTO or at Children's Hospital.
And I don't know if you want to flash back to your second to the last slide there. I think that's where the audio sort of flipped out. But it's a good point to say, I don't think there are many resources that have both adult and pediatric patients that the published reports I've seen. I haven't seen all of them, of course. So it's an interesting thing to look at that. Yeah, it might be this one, Adrie, where I think the audio is lost. I don't know if you want to make any just high-level comments about this part of it. Or the previous.
This one or the next one, the retrospective one.
Well, I did the retrospective one.
So please advance one slide. Yeah, so most of the retrospective are really coming from the adult side, just because the patient population in children is much less. I'm not sure TEL has a number of actual patients that came through the children's hospital. Like I said, we're talking about numbers under 100 that we have through that route.
Yeah, it sounds like there are folks interested in the number of children's patients with prospective samples. I see a question from Dr. Zeffler about that.
Yeah,
okay, we don't have those data yet. I don't know if you have it in the Encore data, Adrie.
Yeah, so for prospective, actually, we currently have 25 positive patients from children's and 8 negative. So it's a good number. It's actually
And then Adrie, while we're here, there's been a request to kind of go over the logistics slides. Would you be able to do just a quick run through of that? I think just because of the audio issues.
Okay, we can go back to that.
Thank you.
So just quickly, so this is an initiative both at UC Health and Children's Hospital. We have 2 routes to collect. One, we talked about the prospective specimens where really a patient is consented and we do a research collection, or the waiver of consent, any patient that has a COVID test done. And we retrieve these specimens, the remnant clinical specimens from the clinical lab, and they all flow into the biorepository. And then for processing, especially the research collection ones, we move specimens to HIMSR, and after processing and into the aliquots, they all flow back into the biorepository core facility. We do all our inventory data into Encore. It's merged up to the patient consent, a waiver of consent data, which is also in Encore. And that data can be moved to Health Data Campus. And they are linked up to the clinical data. And then when research requests come in, we can use that data both from Encore and from Health Data Campus to actually provide these research cohorts with associated data. And there are multiple entities involved in this, as shown on the slide. So, but this is the kind of the global process.
I mean, I'm just gonna make one selfish comment, which is one of the things we've been able to do is really utilize existing resources. Health Data Compass, rich, robust resource, linking that to Encore for the sample correlation and association, the biorepository core facility, which is, so I think one of the things we were able to do this quickly was to utilize existing resources.
I think while we're on this slide, there was kind of a question related to redundancy and how we deal with waiver of consent patients once they are consented. So the question is, to avoid redundancy, if a patient is treated clinically at UCH and a biosample is obtained under a waiver of consent clinically and included in the COVID biorepository, is it unnecessary to also send a research blood sample for that patient to the biorepository when that patient is also included in a clinical research study? So when I answer this, I mentioned that it takes a while for the samples to get to pathology and the consented samples are fresher, but Adrie, would you like to address that at all?
Yeah, certainly for the research specimens, they're much more consistently processed, almost in real time. While the clinical lab specimens, as soon as we can get them released, is like 1 day for certain specimen types. And like an EDTA tube, we might still be able to procure viable PBMCs, but lots of specimens might have set a day or several days, so the quality will go down and also the number of analysis that we can do. That's why for the COVIDome, we're really focusing on those prospective collected specimens, which are much more uniformly handled. Doesn't mean that for the clinical lab specimens that you can't do analysis. It's just going to be a different limit, and there's just going to be different parameters that determine the quality of the specimen.
In terms of, I think there was questions about pediatric subjects and samples. Pete Morani chimed in that there have been about 300 COVID positive tests at CHCO. Of that, there are 20 to 30 patients who have been hospitalized, with about 20 patients who have been contingent to the prospective study. So just adding.
Thanks, Pete. That's helpful.
There's a question about dialysis and CRT. Thank you for the presentation regarding bank specimens, bank samples. I didn't see dialysis effluent waste as included for those requiring dialysis therapies. Have these fluids been collected? If so, how? And if not, would it be possible to collect dialysis effluent waste? Most curious about CRRT modalities.
I don't know if you want to just take that one in the general sense. And I would say, we'd love to be able to serve every request. We have had to have some selectivity just to have the core mission sort of fulfill with those. But Adrie, do you want to comment on alternative sample types?
Yeah, I mean, some sample types are challenging to collect in the clinic, both for where they are mostly collected. Like in ICUs, it's very hard to get specimens collected there. Also, some specimens are really high risk to collect, both for the staff in the clinic as for others. So there are some limitation of how things can be handled. Certainly in the beginning that was challenging, still is, but there are maybe now better ways to rethink some of these efforts that in the beginning we really couldn't do. But yeah, these are very, very challenging specimens that come from an environment where we can't really easily go in, and also has risk at collection, and with still shortages of PPE, that's really a main concern for some of these things.
Yeah, thanks, Adrie.
The next question, any plans to collaborate with other state research institutions, for example, CSU?
Yeah, good question. I mean, I think this was started as an institutional effort. We have a natural, as I think all research institutions do, a collaborative sense. I would expect we'd start to see some joint proposals coming in with researchers from different institutions. And Bryan, Haugen, I don't know if we've seen such things already, or joint sort of efforts or requests, so to speak.
No, we just actually have one we'll be reviewing tomorrow that's between us, our campus, and Boulder. And so, but I have not seen outside, we've not seen outside the institution yet. Although there are some where we have our own investigators working with industries, so there have been those that have come in.
Yeah, good point.
I think the next one's a two-part sample that might need a little bit more specifics, but the question is, how long will it be for us to be able to request unique tissue samples? And I think this is the same person that said, any rough numbers of pregnant patients? Thank you. I think the pregnant protocol has just started, so Adrie, are you able to speak to that?
Yeah, currently we don't have any because it's not included to the basic COVID-19 BioBank protocol. We're just opening the other protocol. Hopefully we'll get our first consents by next week, so I really can't say about much how the numbers will go, but it's really just a starting effort.
And this is a follow-up from Charlene Ortiz with the question about community projects. What, if any, input has community overall been involved in any of these projects? This includes folks outside of the medical field.
Yeah, so I'll make a couple comments, then anybody else in the group that would like to chime in can certainly do that. So it's an important question. I just kind of go back to some of the earlier comments of the difficulty in doing some of our normal things in this setting, just in terms of engagement in general, both with the patients, the providers, research, and the larger community. So I think that has been one of the challenges of this time. Yeah, so again, I don't know if anybody else wants to additionally comment his thoughts on that.
And if you have my contact information, is going to be at the end of the slide and on the website. So if you have specific questions about community research involvement, go ahead and email me and we can follow up offline as well. The next question, is there a plan to centrally classify patients based on severity of phenotype and other variables like age, sex, etc., or is this something individual investigators will have to do? That sounds like maybe a Joaquin and a Tele question.
Yeah, I can take that one. I mean, the short answer is yes, there's a plan to do that for standard severity phenotypes that exist, the NIH and WHO scales, for example, as well as things that Matt mentioned earlier, like ICU admission and length of stay. If investigators have, their own clinical phenotypic definitions that they want to use, then they may need to do some degree of additional curation of the variables, but we have all of the raw materials.
Yeah, and I'd like to comment further, Matthew, that if you go, for example, to the trisomy explorer, you can see not only just the impact of having an extra chromosome 21, but you can start filtering the data by various demographics variables. You can see if something's sex specific, you can filter by age, you can filter by different comorbidities affecting either the typical person or the person with Down syndrome. So we envision that the COVIDome Explorer will have the ability to filter by a core set of clinical variables and demographic variables as well to accelerate research, again, in a collaborative open science platform.
And Matt, I don't know if we have one or two more questions, then we can probably make some closing comments here. It might be right.
Yeah, just two that just came in, and then we can start closing comments. The first last one for Adrie, do we have any lung tissue samples?
So we currently have not really procured fresh tissue samples, especially about being unknown about the risk. Most of the procedures that patients go into, they're tested for COVID, and most procedures are delayed if they have to go into surgical schedules. There might some come through if we would have any, and we've seen a few come through that maybe have resolved disease, but it would all be formalin fixed material and currently no fresh material.
And the last question for today, reminder that my contact information is on the webpage and on these slides. So if you have follow-up questions afterwards, please send them my way. The last question, if our tissue is unique, relatively easily obtained, tongue papillae, and we are able to provide our own PPE, how should we proceed appropriately through you?
Yeah, again, I don't, Matt, if you just want to make some general comments. I think there are these general needs, right? I need blood, I need PBMCs or something, which I think we're able to address. There's some more specific things, and maybe Adrie mentioned this too as well. Perhaps those should come into us and we could just help guide what resources there are. It may be in some cases that the work that Tel is doing might be able to help understand your cohort for different, so again, if it's the lung disease or a head and neck disease, to get a sense of how many samples might be out there and so forth. So I still think it might be good to stop in or maybe work through Matt on those sort of questions.
Tom, I just would want to chime in there. An example of that is this issue of bronchoalveolar lavage. And there were a number of investigators who were interested in that, and that's not something we're actively collecting in the biobank. But what we tried to do was connect those investigators to each other so that hopefully there could be a group of people working on that unique specimen.
That's a great example, Bryan. Yeah, I think that actually worked to bring folks together and be unified. Well, I'll make a few comments, but I'll give a second here in case anybody in the panel wanted to make a closing comment or summary statement of any sort. With that, I just say, I think we had over 200 people here for the vast majority of this. We really do hope this was helpful to better understand what's encompassed in the biobank, how to access it. I'm just very proud of the effort here from different groups or resources on campus to come together quickly in a great collaborative sense to provide this resource for the research community. I encourage everybody to use the website for further information. Matt still likes me right now, but I just say reach out to him with your questions because he's a great navigator, understands this very well. I sincerely appreciate the help of each of the panelists, all the people we've listed here, collaborators across the institution. So thanks so much for joining us. Have a wonderful day, and please contact us with further questions. Thank you.